So our next speaker is Dr. Cynthia Bennett. Cynthia received her master's and PhD from Washington State University, where her studies focused on cognition and sensory processes of the most aged colony of macaques in captivity. She has worked in conservation and research in zoos for almost 30 years. She was the former director of zoo animal welfare at Detroit Zoological Society. She is currently associate editor of the Journal of Applied Animal Welfare Science, where I'm happy to work with her.
Thank you.
Cynthia.
Thank you. I think that no welfare conference is complete without a paper on okapi behavior. So whoops, hang on. They have featured in all of them for the last several years. This is an okapi. It is a large, solitary, forest-dwelling ungulate that has a history of not doing well or being particularly sensitive to captive conditions. What they do, one of the reasons or how that's been diagnosed in this particular species is that they have basically predictive and persistent health problems. Reproductive failure is prevalent in the population, and they have a cohort of abnormal repetitive behaviors that they display quite commonly. Interestingly, this cohort is very similar to the same cohort of abnormal repetitive behaviors that are seen in giraffe, the nearest relative to the okapi. So I think the okapi has something to say about giraffe and how we manage them as well. These ARBs, managers should be concerned about ARBs because they tend to increase in situations that are considered to be suboptimal for the animal, and they are associated with sort of the above conditions. This is basically working with this. We had an opportunity to apply for an IMLS grant that was given to the Brookfield Zoo several years ago. And through that grant, we were able to do an extensive multi-institutional study where we did a lot of survey work with the okapi. We collected hormones from them. We collected testosterone from the males, progesterone from the females, and cortisol from all of them through fecal samples. We now analyzed that. But we also wanted to take a very careful look at behavior and how behaviors would be impacted by the intrinsic and extrinsic factors that impact their behavior. The methods were, to say the least, grueling, but I always bemoan the fact that I only ever had 6 animals in my populations when I was studying okapi, but someone did point out that was probably 10% of the worldwide captive population at one point, and I felt better. But in this, we managed to put 53 animals into the study, and I'm going to report on 46 of those. And when does this ever happen? 23 males and 23 females at 18 different zoos across the United States. The observation took quite a long time to accomplish. It was 3 winter sessions and 2 summer sessions. We actually chose to divide it up into those periods of the year when the management of the animals might be most diverse between institutions. There were an even number of northern or cool climate institutions and temperate or hot climate institutions. We collected a lot of data. Our goal was 15 minutes of focal animal observations for every animal in the study throughout the study, which we did not achieve, but we came very, very close to that. If you look at the bottom, we collected almost 4,000 hours of observation. Multiply that by 4, because that's a 15-minute data set. That's a lot of data, which is great because you can go a long way with it. The behavior we observed during this study, we observed activity budget behavior, but we also observed and paid attention to abnormal repetitive behaviors. Data collection protocols were somewhat different. What I'm going to do is talk about pacing mostly, and please forgive me, I call it pacing, but it's really patterned locomotion. That's repetitive, patterned walking in circuits, etc. But the particular 5 ARBs that appear both in okapi and in giraffe quite commonly are head rolls, object licking, mouthing objects, pacing, and tongue activity. It turns out that pacing and head rolling tend to be locomotor stereotypies, while the rest are oral stereotypies and associated with food. What we used for our unit of analysis is basically the average rate per minute of a repetitive behavior by a particular individual in a data set. Okay. Where necessary, we transformed the data. We did all the stuff you're supposed to do when you do the statistics. It would take a really long time to go through all that. But this data analysis is based on stepwise regression followed by model selection, which is, in old psychology parlance, it's a mixed between within model. And we looked at as I said, the environmental impact and the environmental factors impacting 5 key behaviors. Now, I don't actually want you to look at this too carefully, but I want you to get a sense of what you end up with at the end of the day. At the end of the day, when we actually looked at around 40 environmental and management, social and health factors, this table shows you 14 of them. So you can squint all you want, but it turns out to be somewhat of an indecipherable picture, and it takes a lot to make sense of this. And at this point in the data is where we decided that we needed to put the Okapi back into the data and start looking at what Okapi do in nature and what they're doing here and try to make sense of this kind of big, messy picture. One of the findings that came up was that there are sex differences in 2 of the stereotypies. Males pace more than females. They pace more than females in the summer, in the winter, wherever they are. And females engage in more tongue activity than males. They're significant, and it holds up across any sort of subcategories that we hope to do. The other thing I wanted to take a look at is one of the factors that is most correlated with pacing in males, whether it is summer or winter, is the size of the winter holding. And size makes a difference. Size matters. In the spirit of the thing, I was trying to figure out how to look at how big habitats need to be in order for the Okapi to do well, and we couldn't actually achieve that number. We couldn't find a ceiling or a point where it had to get bigger. We found a floor. We found that things are happening in the smallest habitats, and it has also to do with time. If you look at what the time that they have available to them in the summer, on average, they have 11,000 square feet available to them when they're in their habitats. There are some facilities as big as 30,000 square feet. Some a considerable amount smaller. And they spend around 8 hours, 30% of their day, 33% of their day, in these larger facilities. If you go all the way down to winter, you can see that the average space available to them is considerably smaller. And if you look over at the size of the night quarters, and really, for a lot of these animals, particularly animals that are in cold climates, what they have available to them during the day in the winter is the same as what they have available to them in the night in winter. And you will look and see that some animals are spending 66%, 60 to 66%, in some cases 50, but not many, in 451 square feet. That's on average. Some larger, of course, some smaller. So we're looking at a situation where we have actually a lot of really good sized facilities where the animals seem to be doing pretty well in there, but we also have, but they're spending 30% of their day in the much, much smaller facilities. And that seems to have an impact. And what I wanted to do was take a look at males versus females. The males actually have a very different pattern and amplitude of pacing. And in following the previous speaker, I wanted to look at, she calls it temporal, I call it periodicity. One of the things that I have found in the data, survey data, and in an overall look at large data, even if it's behavioral, is we tend to clump it into 1 or 2 data points, and that doesn't tell me what I need to know. So I want to go back in there and look at data in the way that the animals live their lives. And I was very curious to find out how males and females have partition pacing. It's a huge difference in that. So what we looked at, and if you look at the males, you'll see that they're pacing, and it's on the same scale. I did that on purpose so that you could see kind of the low level of pacing that you see in females, and you can see that it pops up at around 8. What's happening around 8? It pops up again at around 6, or in the summer at around 5. And those are things that Jason was talking about earlier. They were scheduled keeper events. They're either being woken up and fed, or they're being brought in the evening. And the females looks like they're anticipating that, and it actually drops down. If you look at the males, their pacing patterns are very different. They probably do have an element of anticipatory behavior because they too know the schedule. But their pacing is high, remains high, and it starts to actually rise up at around 2 or 3 in the afternoon. And it rises up fairly high. And when we also have preponderance data, so we know the percentage of animals in the population that are doing this type of thing. We also know who does and who doesn't pace. And contrary to some of the data, not everybody paces, but a lot of them do. But they pace at different rates, and perhaps for different reasons. I then went in and started looking at locomotor behavior, because I do know that locomotor behavior, that if you look at the natural history stuff we've been talking about, Okapi are one of those species that actually walk quite a bit, and the males in particular, transit between territories of females, and they're one of those animals that have walking and territoriality, et cetera, as part of their natural history, while the females actually stay put in smaller territories. And if you look, the males start pacing fairly, or walk, or they locomote fairly early, and they continue to locomote throughout the day. and the females go up on locomotion, they stay relatively flat. Well, at around 5 o'clock or 6 o'clock in the evening, when the males are at their peak of locomotion behavior and pretty much at their peak for pacing, that's when we tend to bring them in. And that begged the question for me, is this just extreme anticipatory behavior, or are we looking at an animal that has partitioned its time a little differently than females and might need a different consideration? So I went in and I dug hard for data that was on a 24-hour cycle. There's very little data on any species in zoos that covers the full 24 hours of an animal's life. But there happens to be one study, I did it, I knew it was there, of the animals at the Dallas Zoo. And there were 7 animals in this study, 4 females and 3 males, that we studied for a different reason. And we looked at them 24 hours a day and collected behavioral data. We were actually doing an ACTH challenge for them, but we collected over a 6 to 7 day period. And what we see in the males is that they time shift. Basically, they walk at a very similar rate to females until the early or late afternoon, and then they do most of their locomotion at that time period in the afternoon. The females tend not to do that. You can see that they kind of go up, as you saw in the larger data set, and they stay flat. When they're let inside, it immediately drops down to sort of the baseline level. The males are still walking. They do the same thing for eating. While the males are walking, the females are eating. But they eat later in the day. So it struck me that there's several things that we can pick up from this. One is looking at data as big gross data points might be helpful at letting us know that there's a problem. Looking at data from surveys might be helpful in guiding and directing and having a plan for how to deal with this, but we really do need to take a deeper dive into the data and make our data fit the lives of the animals as they're experiencing them. I can tell you, I feel pretty confident in saying, if you want to decrease pacing across males and females, there's 3 things that you can do. You can make sure there's browse always available, consistent. You can cut down on the discrete number of feeding occurrences. The more times you have a discrete feeding, I feed at 2, 5, and 9, the more pacing you get, and you can increase the size of your night quarters. But both size and time matter. I think that it's important to understand that large habitats cannot offset the impact of significant periods of time in very small quarters. Doesn't matter what it looks like from the front, the animals are having a whole different experience. Sex matters. Shocking, I know. But we tend to manage some species, hoofstock in particular, as if males and females are the same. And in point of fact, males have different life histories than females. And we need to factor that into the management and how we deal with that and how that might affect what we build and how we allocate resources. I think that there's nothing like looking at your animals. I think we need to look at them and look at them critically and have data that actually expresses that. And our data analysis and our data collection strategies need to reflect the way they live their lives, which is 24 hours a day. The thing that concerned me the most when I looked at that is I realized there's very little data on 66% of their lives. We have adjusted our data collection, how our data presents itself, and probably the vast majority of what you're reading in publications that reflect the 8 hours of the day that we're there. So the data that we're looking at in a lot of our journals is more about us and the way we manage our facilities than it is about the animals and how they live their lives. And that's my story. So that's it. As an homage to blatant, horrific anthropomorphism. Everything I needed to know in life, I learned from a hobby.
Thank you, Cynthia. Questions for Dr. Bennett?
Okay.
Well, I'll ask a question.
Sure.
Andrew.
These days there are extraordinary variety of transponders that can track animals all the time. So we don't have to be there, we just hook it up to a computer, we know what they're doing.
Also, there's a lot of video and cameras that are both inexpensive and they're durable. So I think the technology is in a position where we can do those things and should really think about it. Manpower is a hard thing to put there, and there's lots of reasons why you don't want people in an animal's area all the time. You actually probably they need a break from us, just like you need a break. But there's lots of alternate ways to go look at your animal. I'm a little worried about transponders only because you still have to, animals can move or make the transponder think it's moving when they're really moving. So sometimes you still need to get your eyes on them.
Sometimes good is better than perfect.
And that would be better.
Thank you, Cynthia.